Introduction to endosymbiotic theory, a revolutionary concept in cellular evolution. The endosymbiotic theory proposes that certain eukaryotic organelles originated from free-living prokaryotic organisms that were engulfed by another cell. Around 1.5 billion years ago, larger cells began to engulf free-living prokaryotes. Instead of being digested, these prokaryotes survived inside the host cell. Over time, a symbiotic relationship formed, with each organism providing benefits to the other. These prokaryotes eventually evolved into the mitochondria and chloroplasts found in eukaryotic cells today. This relationship was mutually beneficial. The host cell gained efficient energy production and new metabolic capabilities. The prokaryotes received protection and a stable supply of nutrients. The endosymbiotic theory revolutionized our understanding of cellular evolution. It explained the relatively sudden appearance of complex eukaryotic cells in the fossil record. It provided a mechanism for evolutionary leaps and is strongly supported by molecular and genetic evidence. This ancient endosymbiotic event led to the diverse and complex eukaryotic cells we see today in animals, plants, and fungi. To summarize, the endosymbiotic theory states that mitochondria and chloroplasts were once free-living prokaryotes. These prokaryotes were engulfed by host cells but survived to form a mutually beneficial relationship. This process resulted in complex eukaryotic cells and transformed our understanding of evolution. This groundbreaking theory continues to guide our understanding of cell evolution and the increasing complexity of life on Earth. Lynn Margulis was an American evolutionary theorist, biologist, and science author who lived from 1938 to 2011. She received her PhD from the University of California, Berkeley, and later became a distinguished professor, first at Boston University and then at the University of Massachusetts Amherst. Margulis began developing the endosymbiotic theory in the mid-1960s, proposing that certain organelles in eukaryotic cells originated as free-living bacteria. Her theory was revolutionary and challenged the prevailing views of cellular evolution at the time. In 1967, Margulis published her landmark paper on the origin of mitosing cells in the Journal of Theoretical Biology. In this groundbreaking work, she argued that mitochondria evolved from aerobic bacteria and chloroplasts from cyanobacteria that were engulfed by host cells and formed permanent symbiotic relationships. Her paper was initially rejected by 15 different scientific journals, highlighting the resistance to her revolutionary ideas. Despite the revolutionary nature of her ideas, Margulis faced significant skepticism from the scientific community throughout the 1970s. But Margulis persisted, gathering evidence to support her theory, including similarities between organelle DNA and bacterial DNA, shared biochemical pathways, and structural resemblances. By the 1980s, growing scientific evidence began to support Margulis's theory. And with the advent of modern molecular techniques, her predictions were increasingly validated. Today, the endosymbiotic theory is considered a cornerstone of our understanding of cell evolution. For her groundbreaking contributions, Margulis was awarded the National Medal of Science in 1999. Lynn Margulis once said, "If you don't know the right questions to ask, you'll never come up with the right answers." Her willingness to question established paradigms revolutionized our understanding of cellular evolution. The first major endosymbiotic event occurred approximately 2 billion years ago. This event involved two distinct organisms, a larger anaerobic host cell and a smaller aerobic bacterium, likely an alpha proteobacterium. Unlike typical prey, when the larger anaerobic cell engulfed the aerobic bacterium, it did not digest it. Instead, a mutually beneficial relationship developed. The host cell provided nutrients, protection, and a stable environment. In exchange, the bacterium provided efficient energy production through aerobic respiration, producing significantly more ATP than the host could on its own. Over millions of years, the endosymbiotic bacterium gradually evolved into what we now recognize as mitochondria. These organelles retained their own DNA and ribosomes while losing much of their bacterial genome, becoming fully integrated components essential for energy production in nearly all eukaryotic cells. After the first endosymbiotic event created mitochondria, a second major event occurred in the lineage leading to algae and plants. The process began with an early eukaryotic cell that already contained mitochondria from the first endosymbiotic event. A photosynthetic cyanobacterium lived independently in the same environment, capable of converting sunlight into energy. Around 1.5 billion years ago, the eukaryotic cell engulfed the cyanobacterium, but instead of digesting it, established a symbiotic relationship. Over hundreds of millions of years, this relationship evolved as the cyanobacterium gradually lost its independence. The engulfed cyanobacterium gradually transformed into the chloroplast we see in modern plant cells. It retained its photosynthetic ability, but lost many genes and functions. Modern chloroplasts retain key features that reveal their bacterial origin. They have their own circular DNA, a double membrane structure, and still divide independently within the cell. This second endosymbiotic event occurred only in the lineage leading to photosynthetic organisms. It gave rise to algae and plants, creating a major branch in the tree of life. To summarize, the second endosymbiotic event occurred about 1.5 billion years ago. It followed the first endosymbiotic event that created mitochondria and happened only in the lineage that would become photosynthetic. This symbiotic relationship transformed a cyanobacterium into the chloroplast, enabling plants and algae to harness the power of photosynthesis. Beyond the primary endosymbiotic events, evolution continued through even more complex cellular mergers. In primary endosymbiosis, a eukaryotic cell engulfed a photosynthetic bacterium, giving rise to the first plant cells. But the story didn't end there. Secondary endosymbiosis occurred when another eukaryote engulfed a eukaryote that already contained a chloroplast. In this process, a heterotrophic eukaryote engulfed a photosynthetic eukaryote that already contained a chloroplast from primary endosymbiosis. The result of secondary endosymbiosis is a complex plastid with 4 membrane layers: the host cell membrane, the engulfed cell membrane, and the original double membrane from the chloroplast. This process has occurred multiple times in evolutionary history, giving rise to diverse groups of algae with complex plastids. Let's look at some examples of organisms that evolved through secondary endosymbiosis. Euglenoids obtained chloroplasts from green algae, while cryptomonads and haptophytes engulfed red algae. The complexity doesn't end with secondary endosymbiosis. Some organisms underwent a third level of engulfment, called tertiary endosymbiosis. In tertiary endosymbiosis, a eukaryote engulfs another eukaryote that already contains a secondary plastid, adding yet another layer of complexity. Dinoflagellates are perhaps the most remarkable example of complex endosymbiosis. Some species have undergone multiple rounds of endosymbiotic events, resulting in incredibly complex plastids with 5 or more membrane layers. These layered endosymbiotic events help explain the incredible diversity of plastid structures found across different algal groups. Each layer of engulfment added new membrane layers and genetic combinations. By understanding the complex history of secondary and tertiary endosymbiosis, we gain insight into how eukaryotic cells became increasingly complex throughout evolutionary history. The size and structure of organelles provide compelling evidence for the endosymbiotic theory. Mitochondria and chloroplasts are remarkably similar in size to bacteria, typically ranging from 1 to 2 micrometers. Unlike most cellular structures, mitochondria and chloroplasts possess a double membrane. The inner membrane closely resembles bacterial cell membranes in composition and structure. Both mitochondria and chloroplasts divide through binary fission, a process identical to how bacteria reproduce. Unlike other cellular structures, these organelles replicate independently of cell division. To summarize, the structural evidence supporting endosymbiotic theory includes the bacterial-like size of these organelles, their unique double membrane structure, their method of division by binary fission, and their internal organization that resembles their bacterial ancestors. A compelling piece of evidence for the endosymbiotic theory comes from the DNA found in mitochondria and chloroplasts. Both mitochondria and chloroplasts contain their own DNA, completely separate from the nuclear DNA of the cell. The nuclear DNA exists as linear chromosomes. In contrast, mitochondrial DNA is circular, resembling the chromosomes found in bacteria. Similarly, chloroplast DNA is also circular in structure. This circular structure is remarkably similar to the DNA found in free-living bacteria. One of the most significant features of organelle DNA is that it replicates independently from the nuclear DNA of the cell. Organelle DNA encodes some of the proteins needed for the function of mitochondria and chloroplasts. However, over evolutionary time, many genes originally found in the organelles have been transferred to the nuclear genome. The presence of DNA in these organelles, with its bacterial characteristics, provides strong evidence for the endosymbiotic origin of mitochondria and chloroplasts. Ribosomal RNA sequences provide some of the strongest molecular evidence for the endosymbiotic theory. Ribosomal RNA is an essential component of ribosomes in all living organisms. It's highly conserved, meaning it changes very slowly over evolutionary time. This makes it an excellent molecular clock for tracing evolutionary relationships. When scientists compared the ribosomal RNA sequences of mitochondria with various bacteria, they made a remarkable discovery. Mitochondrial ribosomal RNA sequences share striking similarities with alpha proteobacterial RNA. The pattern of nucleotides shows clear homology, with matching regions that indicate a shared evolutionary history. The sequence similarity between mitochondrial and alpha proteobacterial rRNA is approximately 80%, far too high to be coincidental. A similar pattern emerges when we examine chloroplasts and cyanobacteria. Chloroplast ribosomal RNA sequences are remarkably similar to those found in cyanobacteria. The specific patterns of nucleotides show a level of homology that strongly suggests a common ancestry. The similarity between chloroplast and cyanobacterial rRNA is even higher, at approximately 85%, providing compelling evidence for their shared evolutionary history. When scientists construct phylogenetic trees based on ribosomal RNA sequences, the results are revealing. These evolutionary trees place mitochondria closest to alpha proteobacteria and chloroplasts closest to cyanobacteria. These close evolutionary relationships, revealed through ribosomal RNA analysis, provide molecular evidence that mitochondria and chloroplasts originated from free-living bacterial ancestors. Could these molecular similarities be explained by convergent evolution rather than endosymbiosis? While convergent evolution can explain functional similarities, the ribosomal RNA evidence goes far beyond this. The similarities include non-functional regions with no selective pressure, and the precise pattern of shared and unique mutations aligns perfectly with the proposed evolutionary timeline. In summary, ribosomal RNA sequence analysis provides some of the strongest molecular evidence for the endosymbiotic origin of mitochondria and chloroplasts. The extensive similarities to their bacterial counterparts reflect their evolutionary origins as once free-living organisms that became integrated into eukaryotic cells. A key piece of evidence for endosymbiotic theory comes from the complex protein import mechanisms found in mitochondria and chloroplasts. During endosymbiosis, most genes from the bacterial ancestors transferred to the nucleus, but their proteins still need to function in these organelles. Proteins destined for mitochondria and chloroplasts are synthesized on cytosolic ribosomes with specific signal sequences. These signal sequences are specialized amino acid segments that act as address tags, directing proteins to their proper destination organelle. Mitochondria have a sophisticated protein import system consisting of specialized translocase complexes. The TOM complex, or translocase of the outer membrane, recognizes proteins with mitochondrial targeting sequences. After passing through TOM, proteins are transferred to the TIM complex, or translocase of the inner membrane, which guides them to the matrix. Similarly, chloroplasts have developed their own protein import system, with distinct complexes from those in mitochondria. The TOC complex, translocase of the outer chloroplast membrane, recognizes chloroplast transit peptides. After passing through TOC, proteins are guided through the TUT complex, translocase of the inner chloroplast membrane, to reach the stroma. The evolution of these specialized protein import systems was crucial for the successful integration of endosymbionts into the host cell. These complex mechanisms had to evolve after endosymbiosis, enabling gene transfer from organelle to nucleus while maintaining protein function in the organelle. The import systems in mitochondria and chloroplasts differ from bacterial protein transport mechanisms, yet retain some features from their bacterial ancestors. These transport mechanisms were essential for integrating the endosymbiont's metabolism with that of the host cell, a key step in the evolution of the eukaryotic cell. The origin of the eukaryotic nucleus remains one of biology's most intriguing mysteries, with several competing theories proposed over decades of research. Unlike the origins of mitochondria and chloroplasts, which have strong evidence supporting endosymbiotic events, the origin of the nucleus remains more controversial. Let's examine the three major competing theories for how the nucleus originated. The autogenous theory proposes that the nucleus evolved from within the cell through modifications of existing membrane structures like the endoplasmic reticulum. Endosymbiotic theories suggest that the nucleus originated from a symbiotic relationship between two prokaryotic cells. One cell may have been engulfed by another, eventually becoming the nucleus. The archaeal membrane theory proposes that the nuclear membrane evolved from invaginations of the cell membrane of an archaeal cell. These invaginations would have created internal compartments that eventually formed the nucleus. Let's compare these three main theories. Each offers different explanations for the nuclear membrane's origin and is supported by different lines of evidence. Unlike the relatively well-established endosymbiotic origin of mitochondria and chloroplasts, the origin of the nucleus remains controversial. Ongoing research continues to provide new insights, but a definitive answer remains elusive. Who was the original host cell in the endosymbiotic event that led to eukaryotes? For decades, scientists debated whether the host cell was a bacterium or an archaeon. Recent genomic evidence strongly supports the archaeal origin. Genomic analyses have provided key insights into the origins of eukaryotic cells. When we examine different categories of genes, we find a striking pattern. Information processing genes in eukaryotes, those involved in DNA replication, transcription, and translation, show remarkable similarity to archaeal genes, while genes involved in metabolism and energy production more closely resemble those from bacteria. The archaeal host hypothesis suggests a fusion-type origin for eukaryotes, an archaeal cell which contributed most of the information processing machinery, and a bacterial cell which became the mitochondrion and contributed metabolic capabilities, fused to eventually form the eukaryotic cell. The archaeal host hypothesis has several key implications for our understanding of eukaryotic evolution. First, it confirms that eukaryotes likely arose from a fusion event between two distinct cell types. Second, the archaeal host contributed most of the information processing machinery that defines eukaryotic cells. Third, the bacterial endosymbiont, which eventually became the mitochondrion, provided crucial metabolic capabilities. Fourth, complex gene mixing occurred during eukaryotic evolution, with gene transfers in both directions. And finally, this hypothesis elegantly explains the chimeric nature of eukaryotic genomes. The archaeal host hypothesis provides a modern framework for understanding the complex origins of eukaryotic cells, supporting the view that eukaryotes represent a true genomic chimera. Anaerobic eukaryotes live in environments with little to no oxygen, challenging our understanding of mitochondrial evolution. Typical mitochondria perform aerobic respiration, using oxygen to efficiently produce ATP. Many eukaryotes live in environments without oxygen, such as deep-sea sediments, animal digestive tracts, and waterlogged soils. In anaerobic environments, eukaryotes evolved modified mitochondria. Standard mitochondria transform into specialized organelles with different functions. Hydrogenosomes are found in diverse organisms like trichomonads, fungi, and ciliates. They lack an electron transport chain, but produce ATP through substrate-level phosphorylation, generating hydrogen as a byproduct. Mitosomes represent the most reduced form of mitochondria. Found in parasites like Giardia and Entamoeba, they've lost the ability to produce ATP, but retain crucial functions like iron-sulfur cluster assembly. Despite their adaptations, both hydrogenosomes and mitosomes still show clear evidence of their endosymbiotic bacterial origin. Many hydrogenosomes retain the characteristic double membrane structure of mitochondria, reflecting their endosymbiotic past. Both organelles maintain protein import machinery similar to conventional mitochondria, using mechanisms derived from the original bacterial ancestor. Some hydrogenosomes even retain small remnants of DNA, providing direct genetic evidence of their bacterial origin. The existence of these modified mitochondria across diverse anaerobic lineages supports the theory that all eukaryotes share a common ancestor that already contained mitochondria. These findings have profound implications. All eukaryotes, including anaerobes, evolved from aerobic ancestors with mitochondria. There's no evidence for eukaryotes that never had mitochondria. These anaerobic adaptations are secondary modifications of an originally aerobic endosymbiont, supporting the universality of mitochondrial endosymbiosis in the evolution of all eukaryotic cells. Plastids, including chloroplasts, originated through endosymbiotic events where photosynthetic bacteria were engulfed by eukaryotic cells. Primary plastids are found in three major lineages of photosynthetic eukaryotes: green algae in plants, red algae, and glaucophytes. Secondary endosymbiosis occurs when a eukaryote that already contains a primary plastid is engulfed by another eukaryotic cell. This process of secondary and even tertiary endosymbiosis has led to remarkable diversity in algal lineages, each with distinctive plastids. Plastids perform functions beyond photosynthesis, including pigment production, lipid synthesis, and storage of various molecules. The evolution of plastids has created remarkable diversity in pigmentation, giving us the colorful world of plants and algae. Plastid evolution through endosymbiosis has occurred multiple times over the last 1.5 billion years, creating the photosynthetic diversity we see today. Organelles like mitochondria and chloroplasts evolved from free-living bacteria through endosymbiosis. During their evolution into organelles, these former bacteria underwent massive genome reduction. This genome reduction occurred through two main mechanisms: gene transfer to the nucleus and outright gene loss. Gene transfer occurs when DNA from the organelle moves to the nucleus, where it's integrated into the nuclear genome. Several selective pressures drove this genome reduction. One major pressure was the need to avoid genetic redundancy. Centralizing genetic control in the nucleus allows the cell to coordinate gene expression more efficiently. Despite extensive genome reduction, organelles still retain some genes. These are often essential for their function or difficult to relocate. Genome reduction in organelles represents a major evolutionary adaptation that has allowed the integration of former free-living bacteria into eukaryotic cells. During the evolution of eukaryotic cells, a remarkable process occurred where genes from endosymbiotic organelles were transferred to the host cell nucleus. This process, known as endosymbiotic gene transfer, or EGT, was crucial for the integration of the endosymbiont and host genomes. Over evolutionary time, DNA fragments from mitochondria and chloroplasts escaped their organelles and became incorporated into the nuclear genome. Several mechanisms likely contributed to gene transfer. Direct DNA transfer occurs when organelle DNA fragments are released during breakdown and enter the nucleus. RNA intermediates can also play a role, where organelle mRNA is reverse transcribed and then integrated into nuclear DNA. In some cases, bulk DNA transfer occurs, where large segments of organelle genome move to the nucleus in single events. As genes moved to the nucleus, a crucial innovation was needed, targeting sequences. These short peptide segments at the beginning of proteins act as address labels. These targeting sequences direct proteins back to their appropriate organelles after being synthesized by ribosomes in the cytoplasm. The extent of gene transfer is remarkable. In humans, approximately 1,500 genes of mitochondrial origin now reside in the nucleus. In plants, around 4,500 genes of chloroplast origin have been transferred to the nuclear genome. Meanwhile, human mitochondria retain only 37 genes, a tiny fraction of their bacterial ancestors' genome. Gene transfer provided several evolutionary advantages. Nuclear DNA is better protected from the reactive oxygen species produced in mitochondria. The nucleus offers more sophisticated gene regulation mechanisms, allowing for better control of gene expression. Nuclear genes can undergo sexual recombination, increasing genetic diversity and adaptive potential. Transfer decreases the risk of harmful mutations in organelles, where DNA repair mechanisms are more limited. Finally, centralized genetic control in the nucleus allows for coordinated regulation of cellular functions. The transfer of genes from organelles to the nucleus was a critical step in the integration of endosymbiont and host genomes. This genomic integration transformed what began as separate organisms into a single, unified entity with shared genetic interests. It represents one of the most profound examples of genetic integration in the history of life, fundamentally reshaping the eukaryotic cell. Despite extensive gene transfer to the nucleus over evolutionary time, mitochondria and chloroplasts have retained some of their own genetic material. This raises an important question. If most organelle genes have been transferred to the nucleus, why do any genes remain in these organelles? The most widely accepted explanation is the redox regulation hypothesis. This hypothesis suggests that some genes must remain within the organelle to allow rapid local regulation of electron transport processes. Both mitochondria and chloroplasts contain electron transport chains, complex protein systems that manage the flow of electrons and energy production. Keeping these genes in the organelle allows for rapid local regulation in response to changing redox conditions. This direct connection allows organelle DNA to respond instantly to the electron transport status, quickly adjusting protein production to maintain balance. Maintaining redox balance is crucial for cellular health. If redox reactions become imbalanced, damaging reactive oxygen species can form. These imbalances can lead to oxidative damage, causing protein dysfunction, DNA mutations, lipid peroxidation, and even cell death. In summary, the redox regulation hypothesis explains that organelles retain specific genes to maintain rapid local control over electron transport processes. This local control is crucial for preventing potentially dangerous redox imbalances and maintaining efficient cellular energy production. The hydrogen hypothesis provides an alternative explanation for the origin of mitochondria through endosymbiosis. Proposed by William Martin and Miklos Miller in 1998, this theory suggests that the initial endosymbiotic relationship was based on hydrogen exchange between two distinct organisms. The metabolic foundation of this hypothesis centers on the hydrogen dependency of the archaeal host. Early archaea required hydrogen for their metabolism, while certain bacteria produced hydrogen as a waste product from fermentation. This created a perfect syntrophic relationship. The archaeon, likely a methanogen, consumed hydrogen produced by the fermenting bacterium. This metabolic relationship gradually evolved from external hydrogen exchange to full endosymbiosis through several steps. First, the two organisms exchanged hydrogen externally. Then, they developed a closer physical association to improve the efficiency of this exchange. Eventually, the archaeal host engulfed the bacterial partner, internalizing the hydrogen source. Finally, gene transfer occurred from the endosymbiont to the host. The hydrogen hypothesis elegantly explains how the initial metabolic dependency led to a permanent cellular integration. As this relationship became permanent, the engulfed bacterium evolved into what we now recognize as the mitochondrion. The hydrogen hypothesis has several important implications for our understanding of early cellular evolution. It suggests that eukaryotes originated in anaerobic environments and that mitochondria evolved from bacteria specifically involved in hydrogen metabolism. Most importantly, it provides testable predictions that can be examined through comparative genomics and molecular biology. The hydrogen hypothesis provides a compelling explanation for how metabolic dependencies could have driven the evolution of complex cellular structures through endosymbiosis. The syntrophy hypothesis offers an alternative model for understanding the origin of eukaryotes. Syntrophy describes a nutritional relationship where two organisms collaborate to metabolize substances that neither can process alone. According to this hypothesis, the relationship between the archaeal host and bacterial partner began with metabolic complementarity and nutrient exchange occurring at the cell surface. Unlike the hydrogen hypothesis, which proposes immediate internalization, the syntrophy hypothesis suggests that the relationship was established first, with the physical incorporation happening later. The key features of the syntrophy hypothesis include metabolic complementarity, gradual physical association, and mutual benefits established before internalization. Let's compare the syntrophy hypothesis with the hydrogen hypothesis we discussed earlier. While the syntrophy hypothesis proposes diverse metabolic interactions at the cell surface before internalization, the hydrogen hypothesis focuses specifically on hydrogen and carbon dioxide metabolism with immediate advantages from internalization. The syntrophy model suggests multiple possible metabolites being exchanged and a gradual engulfment process, while the hydrogen hypothesis proposes a more specific exchange of hydrogen and carbon dioxide, with endosymbiosis occurring in one step. Both hypotheses are supported by different lines of evidence. The syntrophy hypothesis is supported by the diversity of metabolic pathways in archaea and observed syntrophic relationships in nature. Meanwhile, the hydrogen hypothesis is supported by conserved hydrogen metabolism genes, the ability of anaerobic mitochondria to produce hydrogen, and it explains the retention of certain genes in mitochondria. Current scientific thinking suggests that both hypotheses may be valid and could represent different evolutionary pathways that led to the development of eukaryotic cells. Modern organisms exhibit various forms of endosymbiosis that provide insights into how organelles evolved. The aphid-Buchnera relationship is one of the best studied examples of obligate endosymbiosis. Aphids house Buchnera bacteria in specialized cells called bacteriocytes. These bacteria synthesize essential amino acids that aphids cannot produce themselves. After millions of years of coevolution, Buchnera has lost 80% of its ancestral genome and cannot survive outside its host. Coral polyps form symbiotic relationships with photosynthetic algae called zooxanthellae. The algae provide photosynthetic products to their coral hosts, while the coral provides protection and nutrients. This mutualistic relationship can break down under stress, resulting in coral bleaching. Paramecium can harbor bacterial endosymbionts such as Caetabacter, known as killer bacteria. These bacteria produce toxic particles that kill sensitive paramecium strains, giving the host a competitive advantage. The bacteria cannot complete their life cycle outside the host and are transmitted to offspring. Pollinella chromatophora represents a recent endosymbiotic event that occurred about 60 million years ago. This amoeba acquired photosynthetic organelles called chromatophores from cyanobacteria. Unlike other algae, whose plastids evolved over 1 billion years ago, Pollinella's endosymbiotic event is much more recent, providing a glimpse into early stages of organelle evolution. These modern examples of endosymbiosis provide valuable insights into how ancient symbiotic relationships led to the evolution of organelles. We can view endosymbiosis as a continuum from free-living organisms to fully integrated organelles. Modern examples represent different points along this evolutionary pathway. By studying these modern relationships, scientists can better understand the processes that shape the complex cells found in eukaryotes today. These living examples serve as windows into the past, illuminating the evolutionary processes that led to the diverse cellular structures we see today. Despite strong evidence supporting the endosymbiotic theory, it has faced several challenges throughout its history. When Lynn Margulis first championed this theory in the 1960s, it was largely dismissed by the scientific community. Critics pointed to the lack of observable endosymbiotic events and insufficient fossil evidence. Today, scientists still debate specific aspects of the theory. Some mitochondrial genes don't match expected bacterial patterns. Chloroplast photosynthetic mechanisms show unexpected variations, and the timing of nuclear development remains contested. Several alternative explanations have been proposed. The direct filiation theory suggests organelles evolved from membrane invaginations rather than from free-living organisms. The autogenous model proposes organelles developed from the host cell's own genome. Some researchers have even suggested viral integration as a source for organelle development. The scientific community responded to these challenges with additional research. Expanded genomic sequencing confirmed endosymbiotic signatures in organelle DNA. Comparative studies using advanced techniques revealed phylogenetic relationships that further supported endosymbiosis. Laboratory models demonstrated the mechanisms of gene transfer and integration that explained anomalies in the data. Ultimately, these challenges have strengthened rather than weakened the endosymbiotic theory. As scientists addressed each criticism with more precise research, the theory evolved to include more detailed mechanisms. It now integrates with other evolutionary theories and has gained broader scientific acceptance through the incorporation of genomic evidence. By responding to challenges with rigorous research, scientists have transformed the endosymbiotic theory into a more robust explanation for the origin of eukaryotic cells. Endosymbiotic theory provides crucial insights into one of biology's most profound questions. How did complex eukaryotic cells evolve from simple prokaryotic ancestors? The transition from prokaryotes to eukaryotes represents one of the most significant leaps in cellular complexity in evolutionary history. Endosymbiotic theory provides a powerful explanation for how simple cells acquired the complex features we observe in eukaryotes today. Let's examine the specific mechanisms by which endosymbiosis increased cellular complexity. The acquisition of mitochondria through endosymbiosis provided eukaryotic cells with specialized organelles for energy production. Endosymbiosis also introduced cellular compartmentalization, which provided significant evolutionary advantages. Perhaps most significantly, endosymbiosis offered a mechanism for rapid evolutionary advancement, allowing cells to acquire pre-evolved metabolic systems. By incorporating free-living bacteria, early eukaryotic ancestors gained established metabolic pathways and genetic systems in single evolutionary events, rather than having to evolve these complex systems independently. Endosymbiosis has transformed our understanding of evolutionary relationships. Traditionally, the tree of life was visualized as a simple branching structure with three main domains. In this model, species evolved in a linear hierarchical pattern from a universal common ancestor. However, endosymbiotic events and horizontal gene transfer complicate this neat branching pattern. Mitochondria originated from bacteria that were engulfed by an archaeal host. Similarly, chloroplasts came from cyanobacteria. In addition, horizontal gene transfer allows genetic material to move between distantly related organisms outside of parent-to-offspring inheritance. These processes mean that instead of a simple tree, life's evolutionary history is better represented as a complex web or network. This is especially evident at the base of the eukaryotic lineage, where multiple endosymbiotic events and gene transfers shaped early cell evolution. The modern eukaryotic cell represents a chimera of genes and organelles from multiple lineages, with an archaeal host, bacterial-derived mitochondria, and in plants, cyanobacterial-derived chloroplasts. The web of life concept better captures these complex evolutionary relationships, challenging the traditional tree-like model, especially at critical evolutionary transitions. The endosymbiotic theory has become a cornerstone of modern biology. Over decades, this theory has profoundly influenced evolutionary biology, genomics, and ecological studies. It represents one of the most significant paradigm shifts in our understanding of how complex life evolved. Despite its widespread acceptance, several important questions remain active areas of research. The endosymbiotic theory demonstrates the extraordinary power of symbiotic relationships in driving major evolutionary innovations. Future research will continue to explore new endosymbiotic events, examine understudied organisms, and investigate the molecular mechanisms that allow host and symbiont to integrate. The endosymbiotic theory remains one of biology's most profound insights into how complex life evolved through cooperation rather than just competition.